So please come in. We'll start. I'm Paul Lu. I am the Executive Secretary of the 4 per Mil Initiative. And I'm pleased to be for you today I would say, not the master of ceremony, but the people who will just come and sometime speak and let the floor to the other one. So we have a very, very dense day today, starting at 9, finishing maybe 19, 20, 21. It will depend on you. And it will depend on all our speakers. So welcome to the second day of the conference. And this day will offer the opportunity to deep dive into different pathway and solution related to soil organic carbon in temperate climate country. We'll have a plenary session to start. It's always a good way to start things. And then there will be breakout sessions. But not only one round, we'll have 3 rounds of breakout sessions. So I will ask you all to be disciplined and just try to look at the clock, because if the first breakout session is lasting too long, then the 2 other sessions will be shorter. So give a chance to the other speaker and the other people to have an exchange in all the outbreak session. So at the end, we'll have a joint harvesting plenary in the afternoon, collecting all the information and the data that and the discussion that you will have all together. And we'll have a closing panel, which aim to discuss the priorities moving forward. And then finally, there will be a closing speech, or just try to make a synthesis. And then we will have a farewell cocktail before we close this part of the conference. And of course, I remember you that tomorrow there will be the infield trips. So we'll start this first session of the day. We all know, and it was recalled to us yesterday, we had this speech by Axel Don yesterday, but not only him. Soil are not just a static carbon sink. It's not only dust that we are stepping on. Carbon sequestration and storage are also provided by soil, but they are the result of a sensitive interplay of microorganisms. And this interplay can be easily disturbed, but it can also be fostered, hopefully, by external conditions and activities. This is why we are going to explore this in this session, focusing on the influences of environmental factors as well as anthropogenic factors like fertilization, for instance, in the first 2 presentations, before we will hear about how soil health and soil biology are reflected in agronomic advisory practices in Germany. So, of course, at the end of this session, you will have the opportunity to ask a question to the speaker, and feel free to do so. So I will also ask the speaker to respect the timing, just to be able to get some question at the end. So for this session, we'll have 3 speakers. We'll have Professor Dr. Francesca Cotrufo. You are professor at Colorado State University in the States, and you are a professor in crop and soil science, and you're a specialist of the soil organic matter dynamic. So we will give you the floor to speak about the control of POMs and MOMs. When I see that the first time in my life, I said, what's POMs and MOMs? But you will explain that to us. But this is particularly organic matter, and mineral-associated organic matter. So, Professor, the floor is yours.
It's a pleasure to be here, and also a pleasure to be back in Berlin after, I think, more than 20 years that I was missing. So I look forward to see more of the city as well. I would actually start with a little bit of a provocation. I believe that we have definitely, we need to move beyond carbon sequestration and think more generally about carbon soil stewardship, soil carbon, but more than anything, soil health stewardship. We know that we cannot continue agriculture and management as we have done because we have significantly shrunk the amount of soil organic carbon, and therefore we need to change our management. And first of all, we need to increase the inputs, but also reduce the disturbance so that we can, at the same time, increase the pool of organic matter and destabilisation. And by doing that, we will increase soil structure and will provide more opportunity for water infiltration and retention. And all of this together will promote the soil biodiversity and functioning and bring carbon back into the atmosphere. We do need the soil to respire. We do need the soil to be active. We said the importance of the microbe. We cannot think that we need to shovel carbon in the soil and try to keep it there. And so also because by doing that, that's when we'll start regenerating natural fertility and trying to get away from the massive use of fertilizer. And by doing that, we will increase soil input and try to move the soil to a more resilient system. So that's a systemic change that we are trying to do, and so we don't have to use a carbon focus only. And the nice thing of having been done this work in the, there are many things for which my generation actually feel very frustrated these days, but fortunately the one of us who worked on soil organic matter can be proud of the fact that we have significantly advanced our understanding of soil organic matter and now have a much broader range of concepts that we know are important to determine it. And those concepts are synthesized in this complex picture that, with a former student of mine, we worked in a review last year. And basically, we know now that it's important to see in which form plant material come into the soil, whether they're structural or soluble, and their stoichiometry, and of course, if the soils have the matrix potential to stabilize the carbon. But more than anything, we have the microbes at the center of our story, and that's where actually I will close today, and was important that the introduction pointed to the role of microbes in all these processes. But if you look at these little symbols here, we actually, while we have all these hypotheses and understanding, some of them have some empirical support, but not all of them. And even worse, not much of that is currently incorporated in our process-based models that we use to predict these things. And so there is still a lot that we need to do in order to have in the hands of producer, in the hands of policy, tools that they can use for decision making. At the base of this understanding, there is also the concept now that can help us advance understanding further, that actually soil organic matter is not one thing. We know when I said about the fact that it's very important to determine whether plant inputs come as structural or soluble, we know that because there are two pathways in which soil organic matter forms. One's from the structures, so I'm talking about the more lignified plant materials that mostly enter into the soil from fragmentation and the help of soil fauna that incorporate them, and they are very important for aggregate formation and structure and water retention. And this is the particulate organic matter who's protected only by aggregation, and therefore its persistence is very much dependent on the activity of microbes and their capacity to produce enzymes. The nice thing about POM is that it's mostly being made of structure as relatively less nitrogen in it, and so it's a way also to retain carbon in the soil with the low nitrogen, with the high nitrogen efficiency. On the other hand, the mineral-associated organic matter is formed mostly from the soluble inputs, so leachate from plant residues, as well as the root exudates we know now are the major contribution to mineral-associated organic matter formation, either directly or after microbial transformation. The myome has a lot of nitrogen in it, and so it's a relatively inefficient form of storing carbon in soil, but it's relatively more protected because it's mostly binded to minerals, but a lot of it also exchanged through organo-organo interactions. And so at the base of this now, to understand what we need to understand. It's not just what control organic matter, but really what control POM and MEOM. And so this is the focus of what I'll be talking today. And I always like to, when I have a question, I always address it with the conceptual framework. It really helped me guiding my research and my studies. And so when I came to try and understand what control POM and MEOM dynamics, I came up with this framework that, one of my friends is very good in branding, and so they take the in and out. I don't know how many of you have been in the US. There is a hamburger company that is called the In and Out. But anyway, so this framework basically puts the accent on the fact that we are talking about biogeochemistry. And so everything, it's always the net impact of input and output. And if we want to increase organic matter, it's not a matter of stopping output. It's about having them both work but in balance, or at least in excess of the input, so that we can store more carbon. But depending on whether a system is input and output balance, you would have a different contribution of microbial transformation to the plant input, and therefore a different balance between the POM and the MEOM. And so my hypothesis was that in systems where microbial activity is inhibited more than photosynthesis or plant growth, you would have a system where you have more POM than MEOM and an immobilization of the nitrogen in the POM. And that is expected to be in cold and waterlogged system. Whereas where it's dry, you would expect the microbial limitation to be in excess of the microbial limitation to exceed plant limitation, and therefore the plant input to be more limited than microbial activity, and therefore you would have the microbes transforming all of the plant input and the majority of the organic matter to be stored in the MEOM, and but then you have an open nitrogen cycle, lower productivity, and low carbon. And so our goal for health is really to try and see how we can move this system to a more balanced thing. Of course, the deep soil is by definition input limited, and so we would expect that to be more controlled by the minerals in those soils. We are having our time testing this hypothesis because of lack of data. The first attempt was from a former student of mine, now actually unemployed in Haute, that did a synthesis of global data, and she actually demonstrated and in a way support Axel's point that MEOM is both limited by inputs that are either indirectly controlled by climate or directly controlled also because, as I said, it's mostly leachate, and so precipitation stimulate MEOM formation, and of course by the availability of minerals to stabilize. But on the other hand, what was very interesting is that the POC doesn't seem to be directly related to input because it's not controlled, is not protected. And so it's really the control on decomposition and microbial activity that allow POM to accumulate. And so that brings us to give a little bit deeper dive into the controls on microbial activity, and that seems to be so important in controlling soil organic matter formation. And so when I started thinking about what controls microbial activity and carbon output from the soil, we can really think of three major controls. One is when microbes are inhibited in their activity because they don't have oxygen or because the conditions are too cold. In those conditions, as I said before, they actually don't decompose much of the soil organic matter that come in from plant, and therefore it's mostly accumulated as POM. And that's the situation that we have in peatlands. A lot of carbon in the soil, but this carbon is very vulnerable because if we drain a wetland or if we warm a jelly soil, those microbes become active and can access that situation. But this is POM that can stay for centuries to millennia. So my other point is let's not label organic matter by the turnover time, because today there are the same amount, there are atoms of carbon with the same age for completely different reasons, and they will respond differently both to climate change and management. And so it's better that we model the carbon for the mechanism that retain them in there rather than for their turnover times, because as I show in this picture, the carbon can be in soil for the same amount of time for very different reasons. Because on the other hand, instead, there is microbial that actually cannot access their resource. Those are microbes in which the carbon is stabilized by mineral association, in particularly also to oxidize with iron and aluminum, or cation bridging. They can be in very, very stable microaggregates. Those carbon typically have already been processed by microbes. And so in these soils, we have lost a significant amount of carbon. We hardly have any POM, and the majority of the carbon is now in the mineral form. It's less vulnerable, it's very nitrogen rich, and that can also be stabilized. But that's a completely different form, and maybe climate change will make that less vulnerable. And of course, there is all the situation in the middle where microbes are active, can access their resources, and this is the amount of energy, nutrient, and moisture that control them. And so let's stay more in the efficiency by which they control that transformation. And what I'm saying basically reflects in how we see the distribution of meom and POM. This is a slide from the Lucas soils in 2015. We analyzed them. We are now analyzing the latest sampling. And so we'll see how the situation has changed. But basically, if you look at it, the meom is pretty stable. There isn't that much change in the meom pool. What we have lost from soils is POM. And we see that also when we look at the global soils. Basically, the proportion of meom in the carbon has really increased in the agricultural soils with respect to the natural system. And that has to make us think, because everybody now seems to pursue this meom accumulation. In my mind, we actually should pursue POM accumulation if we want more health and more of that stewardship that we were talking before. And when we look at what happened with regenerative agriculture, it's actually hard to accumulate POM. These are long-term experiments in the US where we actually see a significant amount of carbon accumulated after many years in some of the trials, but it's mostly in the meom fraction. We have very little, with some exception, we have very little accumulation in POM. And POM seems to be responding annually. If a crop puts more residues one year, the POM goes up, but it doesn't respond to multiple years of addition, showing that its turnover is actually completely a pair with input. And so if we want to accumulate POM in agriculture, even with no-till, we need to be thinking about something else. And I believe that we need to be thinking about more perennial roots, more mycorrhizal, more structure, more aggregation that can further protect that. But now, stepping back to the idea of the input and output control, there is also something else that is very important to consider, and that the input-output balance that we were talking about, that here I try to get at that with the ratio of soil organic carbon versus annual MPP, which is an indicator of how much carbon is staying versus how much is the input. We actually see, and these are data from the NRCS, we fractionated the NRCS soil survey, and therefore we have data for all across the US. And what we see is that in arid system, actually it is confirmed that they are more input limited. And so when we think about regenerating soils, it's very important that we start distinguishing what is an arid system versus a humid system, because we might be wanting to do completely different things. It's a very complicated structural equation model figure, but the one thing I want to pose your accent is that there is no link between MPP and particulate or MEOC in humid system, very strong in arid system. In humid, the mechanism of stabilization are by far more important. And so that's need to start driving our decision making when it comes to how can we steward soil and carbon in soils and soil health. We need to really incorporate aridity in our thinking, because we might need to be doing completely different things if we are managing an arid system versus a wet system. And one thing that so far we have ignored, in particularly, I work in Colorado, I deal with a lot of arid soils that have a lot of carbonates. And our first thing is, let's acidify the carbonates like they are our enemies in the lab. But actually it's coming to be very important, the interaction between organic and inorganic carbon in the dynamics in sequestration and soil health. So that's one other thing that I want to point out, is that we shouldn't just be focusing on organic matter, but on interaction between organic and inorganic in arid system, and also on the potential to sequester more carbon by pumping the inorganic carbon out of the system in wetlands. But up to now, I've talked more of a large scale, but there are also some important control at low scale and at the micro scale. And in particular, I think that one, I've been one of the person that said, let's stop thinking about aggregates and let's look at the primary fraction of POM and MEOM. But actually, as Axel was saying, I always like to think more and critically about my own point. And now that we know enough about POM and MEOM, I think it's time to go back to understand how aggregation modify their dynamics, and also, I think, the microbe. And so it's important. We have started looking at the MEOM that is free or that is inside microaggregates. And what we see is that while it's only slightly enriched in 13C and only slightly enriched in nitrogen, has a much higher organic matter density. And when we look at the spectral, it also shows that it's different. And so now we are pursuing a question, and my opinion is that inside the aggregates there is less exchange of the organic matter and more stability and more opportunity for the microbial necromas to stabilize. So that's an area of research that I think we should be looking now more closely. And I want to finish with the microbial drivers. I started with that. It's key. Now we are seeing that actually who's there matter, and we have some winners in controlling the MEOM formation. And when we add microbiome to our models, finally we start having some good R-square. Like the SEM I showed before explained maybe 56% of the variability. When we put the microbiome in our model now, we can go up to even 90, so over 90. So I think that starting looking more closely at the microbe is important, but also not just who's there, but what they do. We go at that question both with some individual-based model that, for example, showed how the capacity of microbes to produce enzyme is key in the destabilisation of POM and the formation of mineral-associated organic matter. But also we are now doing some very mechanistic laboratory incubation in collaboration with OAT to understand what type of microbial traits can affect these processes, and also regional scale in the Corn Belt. We have now a project with Kelly Brighton where we are looking at the microbiome and how that affects POM and MEOM. And our final scope is trying to put all this understanding in our model. We built a model in particular to have a structure that can grow with the understanding. And so as, for example, we are going to have more understanding of the microbial pool, we can start putting some control here. We have different depth. We are developing management and grazing management. So this is really a work in progress. And the nice thing is that we have built a consortium around that, and so it's free for NGOs and for scientists. So we want to create a platform where all of us can work together and increase our understanding, and a company can join and have a transparent access to data and model. My university required that I disclose the fact that I do have a company that actually analyzes organic matter in the soil. These days, I don't know if I'm good or not, but I talk about my research, but they are the guys that do the work. The top are the MEMS modeling team, and the bottom are my lab, Soil Innovation Lab team. And of course, I couldn't do this without all the funding to my lab. Thank you.
Thank you very much, Professor. Well, it's a very strong dose of science in the morning to start. We have the eyes and the brain full of beautiful slides, but we need to at least 1 week to digest all of that. I don't know if it will be pom or mom, but in my stomach, but it will take time. Okay, thank you very much. So we'll have another presentation now on the effect of different fertilization practices on soil health with Professor Dr. Monika Chromnowska. Yeah, I forget the knee, Niska. Sorry, Monika. And so we'll be a little bit more in the agricultural practices and how the fertilizer can have an influence. Thank you very much. The floor is yours.
Hello, everybody. Thank you for inviting me. It's great to be here. I was asked to say a few words about the effects of different fertilization practices on soil health. The range of soil health indicators is extensive, so it's a huge challenge to present them within the very short time frame. And I'm going to focus on parameters that are largely influenced by fertilization practices, play a crucial role in maintaining soil health, enhancing crop productivity, ensuring environmental quality, and help to mitigate and to adapt to climate change. As you have probably guessed, I'm referring to the content of soil organic carbon and nitrogen content in the soils. In the European region, low organic carbon content is a key sign of declining of soil health, and excessive amounts of nitrogen described by Mark Sutton as the godfather of pollution is a key indicator of soil health. Crop efficiently use only 50% of nitrogen used in fertilizer used, and 25, sometimes more, % of nitrogen used in fertilizer is lost into the environment. Therefore, in agri-ecosystem, it's crucial to implement proper fertilization practices to maintain optimal levels of carbon and nitrogen compounds in the soil and to minimize their dispersion into the environment. In cooperation with the National Chemical and Agriculture Station, we conducted a large-scale environmental study to identify the effective fertilization practices used on farms that influence the accumulation of organic carbon and nitrogen under Polish environmental conditions. We analyzed soil samples from over 3,000 locations from Polish soils, and it was found out that in Polish agro-environmental conditions, significantly the highest organic carbon accumulations occurred in soils fertilized with manure at the rate 150 kilograms of nitrogen per hectare. It's almost 4 times above the average use of this fertilizer in our country. We also stated that a significant reduction in the content of soil organic carbon was observed with the increasing rate of the crop and pre-crop nitrogen fertilization above 50 kilograms of nitrogen per hectare. Some authors observed that nitrogen fertilizer application enhances carbon accumulation in agriculture ecosystems only when it is supplemented with carbon farming practices such as straw incorporation. We also stated that significantly the highest amounts of ammonium and nitrogen were accumulated in soils fertilized with mineral nitrogen at rates exceeding 150 kilograms and 100 kilograms per hectare applied as respectively to the main crop and pre-crop. Fertilization with manure had no significant effect on the content of ammonium and nitrogen in the soils. We should remember that most of nitrogen in manured soils may be subjected to the immobilization, and its transformation into available to plants forms occurs in longer period of time. At this point, it should be underlined that gross nitrogen balance is one of the best tools for determining nitrogen fertilizer rates. It is calculated as the balance between inputs and outputs of nitrogen in the agri-ecosystem, and it allows for reducing nitrogen surplus, increasing nitrogen use efficiency, reducing nitrogen losses, and lowering fertilizer cost. Therefore, it's a key element of sustainable nitrogen management in agri-ecosystem, combining production, economic, and environmental goals together. In addition to the right rate, we should also pay attention to the right fertilizer source, right time, and right place of its application. Fertilizer work best when the 4 R's stewardship meets the right condition. Therefore, optimization of the soil environment for the favorable transformation of carbon and nitrogen is crucial. In Poland, optimal soil pH is a priority since 40% of our soils are acidic and highly acidic. We should remember that the lowering pH below the certain levels optimal for some certain nutrients could dramatically decrease the efficient nutrient use of other nutrients. And in the case of nitrogen, the optimal rate is between 6 and 8. Some farmers try to compensate for it by increasing fertilizer rates. In our large-scale study, we also established that the decline in soil pH was accompanied by increase in ammonium, nitrogen, and decrease in nitrate concentration, which was undoubtedly connected with the increasing rate of nitrification. We also established that soil pH was one of the most important parameters which influence soil organic carbon content. Statistically significant decrease in soil organic carbon content was observed in acidic and highly acidic soils, and soils with pH above 6.5 accumulated the highest amounts of soil organic carbon. Liming affects not only the quantity but also the quality of humus substances in the soil, which a lot of researchers explain that non-acidic soils favor the transformation of humus substances into relatively stable organomineral complexes that are more difficult to decompose, and on the other hand, greater inputs of post-harvest residue soil inline soil compensate for initial carbon losses caused by increased mineralization. We sometimes forget that for sequestration 10,000 kilograms of carbon, 833 kilograms of nitrogen and 200 kilograms of phosphorus are needed. Moreover, deficiency even of one element influences on the decrease of the nutrient use efficiency of other macro and micronutrients. And the unbalanced fertilization with phosphorus and potassium is another unfavorable factor for carbon sequestration and nitrogen use by crops in Poland. Please note that N/K ratio in mineral fertilizers used in Poland has decreased from 1 to 1.3 to 1 to 0.5 during the last 50 years. Moreover, in Poland, 1/3, sometimes more, in some regions, is characterized by very low and low phosphorus content. In our study, according to farmers, the main barriers in analyzing soils are connected with the time-consuming nature of sampling and testing, high costs, and limited access to advisory support. Access to knowledge and infrastructure for soil health monitoring is essential for stakeholders to understand the effect on soil health. And after our workshops, the increase of soil analysis among farmers increased according to the regional agriculture and chemical station in Lublin. In conclusion, I would like to underline that soil health-friendly fertilization should integrate both environmental and production goals by using precise adjusting of nutrient inputs and optimizing the soil environment for the favorable transformation of carbon and nitrogen. Regular soil testing is essential for monitoring, maintaining and improving key indicators of soil health, as well as optimizing nutrient management. It also prevents both the over-application and under-application of the fertilizers. Thank you.
Thank you, Monica. Thank you very much. And thank you for your conclusion and also one of your last slide. It makes the link directly with the last presentation, I think, with Dr. Konrad Hegenolf. You are policy officer in the Chamber of Agriculture of North Rhine-Westphalia in Germany, and you will specifically present to us the fact to develop an advisory services for soil organic matter buildup. What are the challenges and what are the opportunity? And this will be the relation with the farmers, as Monica has just stressed. We go from science to the farmer, and never forget that the farmers are the one doing the job in the field. Okay, yeah. Konrad,
okay.
Floor is yours.
Thank you, Paul, for the introduction, and thank you for the invitation, Claudia and Samuel and the organizing team. Yeah, as it was mentioned, I'm working at more at the field scale, advisory level. I'm working for the Chamber of Agriculture in North Rhine-Westphalia, Western Germany. We are part of the Humus Klimanet, so we are assisting 28 farms in Western Germany on soil organic matter building measures. And within these first 3 years of the project, we are trying to develop an advisory service, which I want to, or the ideas and concepts we were developing in the recent years, I just want to share. And yeah, I will start with this. I think I don't have to mention it anymore, the organic carbon cycle. But maybe just to emphasize, we're talking about, when we talk about soil organic matter levels, we're talking about steady state equilibriums. And if we want to reach a shift in the steady state equilibrium, we really have to understand the different processes which are involved, especially on the lower part, when carbon cycles throughout the soil. And I have the feeling in day-to-day advisory work, our tools to do so, to assess the site-specific conditions of a soil are very limited. We're still working with standard wet chemical analysis of nutrients, and we do maybe some manual soil structure tests, but that's it. And we really try to get a bit beyond that to, in fact, push the system to a new level. And I think I want to start this talk on this traditional approach of advisory or soil organic matter balancing, which we were focusing on in Germany. It's a method which was developed by the VD Lufa. It's very simplistic, in fact, just looking at soil organic matter reproduction rates based on organic amendments, crop residues on the one hand side, and crop-dependent soil organic matter losses on the other hand. It was actually incorporated, in Germany it was incorporated in the conditionality of the last CAP period, so farmers were obliged to calculate these soil organic matter balances in order to receive payments. And if I talk to farmers, farmers are not very in favor of this approach. The problem is it doesn't really align with what they experience. So they say, I've been doing that since 10 years, the balance is always positive, nothing is happening. As a soil scientist, I would say it's not so surprising because it's a very reductionist approach. We don't think about site-specific soil conditions, climatic conditions. We don't take them into account. But anyhow, starting the work 3 years ago, I said, like, is that really true what farmers tell me, or is that should I stick to that approach or not? So we have soil organic matter monitoring program in North Rhine-Westphalia. It's now in the 15th or 16th year since 2009. And I just plotted like the observed soil organic matter changes throughout this period of 2009 till 2021 against the results of my soil organic matter balance. And you can see the R square is 0.00. So farmer's perspective is actually quite correct. It really doesn't predict at all what is happening. So then we started within the project, I think the overall project consortium of Humus Klimanetz, but also within Chamber of Agriculture, we have a group of 10 extension services which are working on that project to think about where could we actually intervene, what are concrete management practices, management advices we could give to the farmers. I feel there are high expectations. They think now the experts are coming to the farms and now everything will change. And then you stand there and say, well, what do I tell them now? And we, at the moment, I think we're focusing a lot on maximizing inputs. This is very straightforward, I would say. But we already see that the potential is quite limited. So, of course, cover cropping is easily applicable. It's I would say, a long-hanging fruit. It's easily any farmer can, especially if he is compensated for the additional costs, can implement cover cropping. We work with more complex cover crops mixtures. We try to optimize the cover crops mixtures. We work with additional cover crops in the summer between 2 winter main crops. Then we try to work on crop rotations, so exclude soil organic matter depleting crops in favor of soil organic matter building crops. This is very hard. At the end, potatoes, sugar beets in our region are the cash crops and the farmers are dependent on them. So the willingness to really exclude these from the crop rotation is not very high. And then if we talk about organic amendments, this was explicitly excluded in this project because we say if we apply more organics to one field, that might actually lead to the decrease on another field, and these are leakage effects which we don't want to provoke. Although in a certain extent, especially in my region, we have in the north high livestock production and in the south just arable cropping. To a certain extent, I think it would make sense to redistribute. Anyhow, it's, the overall potential we do see is limited. So let's look at the other side, losses. And essentially, if we talk about minimizing carbon losses, I think we talk about managing priming effects. So priming is defined as increase in soil organic matter turnover due to any management intervention, be it fertilization, be it crop tillage, be it irrigation. And I think this is what Francesca mentioned with managing the POM fraction. So this is the fraction which is easily decomposable. The aggregate stabilized soil organic carbon is actually starting mineralizing when we destroy these aggregates. So it's all about improving soil tillage, reducing tillage intensity when we can, and not provoking priming or soil organic matter turnover when we don't want it. So it's a dynamic system. We need soil organic matter turnover in certain times. An organic farm is dependent on soil organic matter turnover in spring because we need the nitrogen to nourish our crops. But we definitely don't need it in autumn. We don't need it in winter. So it's a way of how to manage these priming effects, which we induce by very many management practices on a day-to-day basis. And then thirdly, like a bit the black box, what is happening in the soil? How can we actually, if I point here to this, how can we divert more of this carbon towards stabilized form, this MAOM fraction, instead of losing it to carbon dioxide? And I think a critical point is, as Axel mentioned yesterday, we depend on the soil microbiome, which is actually converting POM to microbial biomass and afterwards to stabilized organic matter forms. And how to increase carbon use efficiency. We know there are specific conditions, be it temperature, be it moisture, be it pH, which strongly influence the efficiency of the soil microbiome. And so in a way, it's a matter of optimizing the physical conditions at the first glance, but then also chemical conditions. So we also think that, or we know that nutrient limitations are actually also affecting carbon use efficiency. So it's a way of thinking not only on nourishing the plant, but nourishing the soil in an adequate way. So to maybe point out the potential, it's a theoretical potential, it's just theoretical numbers, but carbon use efficiency, those conversion steps from POM to microbial biomass, can range between 20 to 60% according to literature, and this whole process towards the stabilized organic matter is actually more or less in the range of 10 to 30%. We have to mention, normally we're at the lower range of this span, but at least there is the potential to increase, and I think we should think about how doing that. So, yeah, having said that, I think I want to introduce two things. First of all, is our approach towards this improvement of the crop rotation, because I think this is something which is of benefit for the farmers and which could also contribute to our idea of increasing soil organic matter levels. And then I also want to point out, I want to shortly speak about the second topic, carbon use efficiency, how we would like to address this. So with respect to the crop rotational planning, we use a tool which is called NDCEA. It is basically a carbon model. It was developed already in the 90s, I think, in Wageningen. It was a very old-fashioned desktop app which nobody was using anymore. And we had a project a few years ago at Chamber of Agriculture, and we said, actually, it's useful, especially for organic farms, to align nitrogen delivery with crop demand. So essentially, what it does is you just have to give the program the information on your crop rotation, on different fertilizer amendments. These are these blue arrows. Then on crop residue amendments, so is the crop residue remaining or is it taken away from the field? And then it's a simple carbon model which is telling you how carbon levels evolve over time. So in the lower part you can just see that this brown curve is the changes in soil organic matter. And these bar plots are actually showing on a yearly basis the overall balance. Yeah? So the colorful bars are the inputs, and this lower bar, the gray bar, is like a negative bar, which is carbon turnover or loss. If we look at that, we can already see where are we losing strongly, where is it strongly decreasing, and where is the system actually increasing. But more essentially, if we think about this relation or this ratio of carbon to nitrogen, I mean, it's true that it's very different from different carbon fractions, but if we take the overall soil organic matter, it is relatively stable. So at the end, I would say, without additional nitrogen, there's no additional soil organic matter buildup. And on the other hand, we could also say, if we lose nitrogen, we lose soil organic matter. So this system also, you can plot different things. You can also plot mineral nitrogen in the soil, topsoil, subsoil. You can plot cumulative nitrogen mineralization. But this is an interesting graph because it plots leaching losses. So we actually go to the farm and say, okay, look, you have this problem because you're cropping winter wheat, which is developing very lately, which is not taking up sufficient nitrogen after cabbage. Do something about it. Yeah, try to reduce these nitrogen losses, because every kilogram of nitrogen we're losing would mean more or less 10 kilograms of carbon. So this is the way we are approaching this, and it's interesting because it doesn't essentially mean that you have to take crops out of the rotation, but it's just about optimizing the sequence of the crops in order to align carbon and nitrogen fluxes to the demand of the crop rotation, and by that reducing losses. And of course, it's also, or the additional benefit is we don't have we have a more efficient system, reducing losses to the groundwater, reducing maybe also nitrous oxide emissions. The second point is that we have the feeling, we go to the farm, we do like some manual soil diagnosis, and then we come back a year later, we're not really there at the same time. There's a different pre-crop, the field looks totally different. So we're a bit searching for a more objective way of assessing the microbial condition of a soil. And we are actually in this humus project, which is funded by the Soil Mission. And we were looking for soil biological indicators which would be cheap and easy and scalable, and also more or less relevant in this day-to-day advisory service work. And one of the approaches we are taking now is this microresp assay. I don't know if you know it. It's a very simple incubation just in order to quantify basal respiration, so the activity of the microbiome, and also it allows to estimate microbial biomass. And then the interesting thing about that is when we can derive the metabolic quotient, so just the quotient between basal respiration and microbial biomass. And this is an indicator, or it's discussed as indicator for carbon use efficiency. And of course also, if we think about long-term trends, we know microbial carbon and soil organic carbon are highly correlated, but there are some hints that actually propose that if this C-MIC to C-ORG ratio increases, then this might be actually a site where we slowly start building up soil organic matter. So we see that as a short-term indicator for soil organic matter trends, or at least as a hint if we're on a good way or not. We're just starting with this work, so we have established this assay at our lab. It was a big difficulty to establish it based on dry soil samples. We want to use the standard samples farmers are sending to the lab in February, March, or in August. So that was the work of the last 1.5 years, but we were quite successful in doing so, and we think that, we hope that we can offer that to the farmers by a commercial lab by next year. And we're already working on the project farms, especially on these different demonstration sites or pilot experiments on the farms to actually track changes along the way. So to wrap up, what we have in mind for future is that we're doing it partly, but I think not completely. So this advisory service would comprise a baseline evaluation, just standard physical, chemical, and in future biological parameters. We evaluate the current status of soil organic matter level. This is a tool provided by Thünen Institute just to say, are we actually at the average or above or below, to identify fields where we can really do it or have a success. Then we start with soil management, so trying to optimize liming and fertilization, but also tillage. And then the third step would be this crop rotational planning, very strongly focused in cover cropping and also improving the crop rotation based on the NDCA tool. And at the end, we need some, let's say short-term monitoring approach, which allows us to say if we're on the right track before or in a shorter time span than 10 years when our next soil organic matter assay is actually telling us so. So this is our approach to, it's manual and also now lab-based, but should be more direct indication than soil organic matter levels on the long run. Okay, thank you very much.
Thank you, Konrad. So I think maybe I will invite the three speakers to come over here, just for a round of questions. Is there any volunteer, the first one, to ask a question? Everything was clear? No question at all? Ah, yes, thank you, gentlemen. Can we have a mic for this gentleman over there? So we have 10 minutes just to ask some question. Okay, please, go ahead.
Hello. Thank you very much for your presentation. I'm also part of the Humus Klimanet, so I know Konrad. And I have a question for you, but maybe all of you, regarding the priming effects. As I understand, priming effects can also occur when there is additional organic matter added to the system. And I wanted to ask about that, what are the insights on that so far? If we add more organic matter, can it have negative effects on what you were explaining and what we are trying to work towards to increase MAOM in the system, but maybe we have a priming effect that works against it if we are adding soil organic matter. I don't know if I made myself clear like that.
Okay, so you, yes.
I can give a mechanism. Can you hear?
Yes.
Yeah, I can give more mechanistic answer, and then maybe you can do a more practical one. So from all our studies, when you add fresh carbon to soils, you always induce a priming, but the priming is always less than the net difference. So there is always a gain at the end. And so let's not be, taken by the priming as necessarily a negative thing. Sometimes it does result in rejuvenating the soil organic matter, but the net amount of soil organic matter at the end is typically always higher than what it was before. So that's one important component. But it also goes to what we were saying before, and that what also show in those results is that, in particular in humid environment, input are not the answer, are the necessary condition to increase organic matter, but are not the only condition, and it can result in a net zero change if you don't modify management to increase the stabilization. And the other thing is that very important to consider what type of input you put. And in my mind, it's not POM going to microbe, going to meom. That is actually a minor transformation and a minor formation of meom. It's mostly exudates and leachates that go to microbe and meom or straight to meom. And so my suggestion to why the calculation doesn't work is because we don't quantify exudates from plants. And until we have continuous roots and mycorrhizal, and that would be very, very hard to accumulate that POM and accumulate those organic inputs that we put in soils in a humid environment.
Thank you. Konrad, you want to complete?
Yeah, I think when I talk about priming, I'm primarily talking about priming induced by soil tillage and irrigation and weed control in organic agriculture. And with respect to organic fertilization, at the end it depends on the CN ratio. So if you have substances with a very narrow CN ratio or mineral N, then that leads to positive priming, so an increased soil organic matter turnover. The wider the CN ratio is, if you have a compost, a manure, then priming effects are very low. And it's absolutely right. The amount of carbon you add is much higher than that what you lose by priming.
Thank you. Another question? Yes, please.
Thank you. My name is Abhinav. I work at Oath. The common thread I heard through all the talks is the black box of microbiology, right? And how do we, from a research standpoint and a practical standpoint, attack this, unpack this? Because it's so complicated, right? And no solution will fit all. So how should we think about this, moving forward?
So, yeah, we have to start working on quantifying who's there at scale. Unfortunately, and it's not just who's there, but the entire microbiome stuff, which is incredibly expensive. And so it's really looking at it efficiently and cost effective. So the way in which we tackle it is that we are doing a paired analysis of the full microbiome, and we need to send sample to the EMSL lab. So like only the DOE, at least in the US, has this capacity. And then we pair it with our fractionation and all our soil parameters. And we then use models and artificial intelligence to understand the links, so that we are trying to get some traits that we can then put in models and go from there. So we are still at A. There is a ton of science and work, but I'm now convinced that, for many years, we have always think if we do pH, if we do climate, if we do something, we can predict who's there and we can ignore who's there. Now it's obvious that it's not the case. And I think it also depends on the fact that since we have degraded soil so much, we have also degraded the microbiome, and so there are some important components that are not there anymore, and we might need to re-inoculate the soils.
Thank you. Yeah, the lady over there.
Rosa Mosquera from the University of Santiago de Compostela. I'm the coordinator of one of the two subsoil projects at European level from the Soil Mission. We are working to deep soil layers to see how the carbon is established. Preliminary studies carried out in the most extensive agroforestry systems in Europe, which is the HESA, shows that below the tree you have double amount of carbon stocks than far away from the tree, so in open sites. So my question is, with regard to, did you take into account these deep soil layers in your modeling analysis? Thank you.
So, who is answering?
So we have developed a model that has discrete soil layers, and we hope to be able to model the deep soils. When we try to do meta-analysis or structural equation modeling to top and bottom, there are very few data on the deep soil, and so it's very hard to do the same type of analysis we do on the top soils. But yeah, I totally agree with you that are an important component of the system to quantify the stocks. But to be honest, when we look at the changes, unless you start doing introduction of perennials with deep roots, we don't see that many other of our regenerative practices are modifying the deep soil. So I totally agree that we need to quantify deep soils to have an accurate estimate. I'm not convinced that we need to monitor the changes at depth unless in our practice we change and again have very, very deep root systems.
Clear? There's one question over here. Yeah.
Thank you. Ahmed Al-Bitar from National Center for Research France, CNRS. I'm head development of MRV tools. So we've seen quite nice presentation about who's living in this environment, the microbials, and then what they are interacting with in nitrogen. My question was how they are living, in the sense that I am originally a fluid mechanics, and I know that in the soil, people, we say in France, casser les dents, they had broken their teeth on parameterizing the multi-phase flow and thermodynamics in the soil. And scaling this at very large scale is very challenging also. So adding the microbial will be quite impossible to do at very large scale. So I'm wondering, how do you see on the missing processes you're adding, you're neglecting in the water and the energy balance, and how you see it to scale up?
Okay. Ooh.
I have to answer. Yeah, it's a great point. For the microbial component, we are, there is the idea of the parsimony, with modeling. So we are trying to understand what are the traits that are really important to put there, and we are doing it starting with the microbial models and trying to understand what are the microbial parameters that are key to introduce. But we are not quite there yet, and how that is more or less important than adding more. I would love to represent redox better in our soil, to represent a dynamic pH. There are a ton of things that a lot of the process-based model don't do yet, and as an experimentalist, I feel very guilty because a lot of the things that I would like to put into models, when we try, we don't have the data. We don't have dynamic bulk density after tillage, and so there is no way to do tillage right. So there is also a problem. We are spending a ton of effort in quantifying things, but I don't think that we are quite there in quantifying the right things that are important to advance the modeling. And having worked now across the two teams, I'm getting it better. Yeah.
It's very difficult science. All I know is I don't know anything. Arwin.
Thank you very much. Aaron Jones, European Commission. The EU soil strategy has a concept of testing your soils for free, which member states should implement. Can I ask the panel what measures they would encourage people to test for in relation to carbon sequestration? And I should know, but I don't remember at the moment, is there a CEN ISO standard already for POM and MOM analysis?
Okay, we have 1 minute to answer the question.
So, and actually that was also a suggestion to you. I have a huge faith in FTIR. We are about to predict almost everything, even ecosystem, soil health outcomes, or carbon mineralization, or POM percent, MEOM percent, soil carbon. So I think that we need to really develop very strong model, and then we can do FTIR that is relatively cheap and non-destructive, and we can predict the majority of this function in soil. So that would be my suggestion. But yeah, POM needs to be done by density, and MEOM needs to be done by size.
Conrad, element of answer?
Yeah, we were thinking about that, but POM and MAUM is just too time-consuming and costly for routine analysis. So FDR I'm not too familiar with. We are thinking about thermal fractionation. There are some very critical discussions about that, but at least it might be you might be able to differentiate between a labile and stable soil organic matter. And if we add CN ratio or information on the CN ratio of these different fractions, that could be interesting. But we are just starting that, so I cannot really...
Some clue on that?
Because the changes in soil organic matter is rather slow, so one of the quickest, I think, way to check the changes in soil organic carbon, it would be like dissolved organic carbons forms and labile forms of carbon. So it would be, I think, that the quickest way to check, because we have to wait to changes in total organic carbon for years sometimes. So.
Okay, just 1 second.
Yeah, I would like, one thing that we haven't touched on is animal integration. That goes to the fertility and stuff. And I see we have an N equal 9, I think, in my analysis. So there's very, very, very little data, but we see huge improvement when livestock and even chicken are integrated back into the farm. So that's, to me, is one thing that we should do more.
Thank you very much. So a big applaud for all our panel. Thank you. So I think we are quite okay on time at the moment, so we'll try to keep the track. So we will have now a first round of breakout session. They will start at 10:15. So please, if you know where you are going, I mean, just go straight after the end of this session. So there will be, in this room, there will be the session on policy, regulatory, and market-based instruments. In the room number 1, which is the room next door, there will be a session on living labs and soil organic carbon. And in room 2, then you have to go across to the place where we have the coffee break. You will have a session on biochar and circular economy. So we remind you that you can find the whole program at the back of your badges. You have a QR code. You can scan this QR code and you can get the whole program of the day. So if you don't have yet selected the breakout session you want to be in, so please consult the program and select your room very quickly. I just would like also to recall you that in this room over there, we have a small exhibition with some booth and stand. So we encourage you to go there, look at what's there and discuss with the people they are close by. And then also you have the posters. But the posters is more obvious because that's the place where you're having the coffee. So most of the people will look at the poster with your coffee. You are not allowed to take coffee over here, but you start by here before going to the coffee break later. Right. Then after the first round of breakout session, we'll move to the lunch, and then we'll straight go to the second round of breakout session and the third one. Then I recall to you that after the last round, there will be the joint harvest of all this breakout session, and then we will conclude the day. So we're on time. Keep being on time for the rest of the day. And please, now you can spread all over and go to the breakout session. Have a good day and see you soon.